Hello friends, today we are going to study about the process called as ubiquitination. So as we all know, there are so many proteins produced within our body. It is likely that our body may sometimes produce the misfolded or the faulty proteins. So it becomes important to degrade such proteins into smaller amino acids so that they don't cause any harm to us. Okay, and the process of tagging such faulty proteins so that they can be recognized by the proteosomes is called as ubiquitination. For example, this is the misfolded protein and these are some of the healthy proteins. Okay, so how will the proteasome recognize among all of these proteins that this one is the faulty protein? So this has to be tagged by the special proteins called as ubiquitin, and these tagged proteins will be recognized by proteosomes and then degraded into smaller amino acids. So the process of tagging such faulty proteins by the ubiquitin protein is known as ubiquitination. Okay, and the interaction between this protein and the ubiquitin is by a bond known as the isopeptide bond. These are bonded to each other by a isopeptide bond. This we will understand in detail using the molecular diagram of this ubiquitin and protein. Suppose this is the ubiquitin residue, that is this is the lysine. Sorry, this is the glycine, guys, and the another side is the protein residue called as the lysine. This is the lysine. Okay, so the last residue of the protein is the lysine and the last residue of the ubiquitin is the glycine, and this interaction involves the carboxyl C terminal of the glycine, that is a carboxylic group, and the N terminal of the lysine bonded to each other by the isopeptide bond. Okay, now this bonding of glycine and lysine involves the use of three enzymes. Those enzymes are ubiquitin activating enzyme, also called as E1 enzyme. Second is the ubiquitin conjugating enzyme, called as the E2 enzyme. And the third one is the ubiquitin ligase enzyme, also called as the E3 enzyme. Now, in the first step, we use the first enzyme, that is the E1 enzyme. This enzyme involves the activating of ubiquitin protein. Suppose this is the ubiquitin protein we have, and this protein is activated, and this step, the first step, involves the use of ATP. Okay, this is the E1 enzyme. This step involves the use of ATP and release of one molecule of AMP and phosphate groups. Okay, and this forms the E and the ubiquitin complex. So let us see how this bonding happens. Here, this is the E1 enzyme and it has a active site called as cysteine sulfhydryl site. Okay, this is a cysteine sulfhydryl active site of the E1 enzyme, and here the ubiquitin protein comes and binds. Okay, and these both are bonded by a thioester bond. Okay, and the second step involves the ubiquitin conjugating enzyme, that is the E2 enzyme. Okay, so what happens here? The E2 enzyme comes and takes away this ubiquitin enzyme from the E1 enzyme. Sorry, this it takes away the protein, ubiquitin protein from the E1 enzyme and attaches this ubiquitin to the active site of E2 enzyme, that is the cysteine sulfhydryl site of this E2 enzyme by a thioester bond. This also forms a thioester bond. Okay, in the third step, involves the E3 enzyme. So E3 enzyme is attached to the substrate, that is the protein which has to be degraded. Okay, now this E3 enzyme takes away this ubiquitin from this E2 and it takes away the ubiquitin from this E2, and now this ubiquitin is attached to the N terminal of this protein substrate, that is lysine. It has the lysine residue at the last, and this lysine residue is attached to the C terminal of the ubiquitin to the N terminal of the lysine, that is a glycine, and this is the lysine residues. We have here in the last ubiquitin we have the glycine residue, here in the last we have the lysine residue. Okay, these now form the isopeptide bond. Okay, now this is taken away by this and this E3 leaves. Okay, now this forms the substrate, the ubiquitin tagged. Okay, now this ubiquitin is tagged by this protein is tagged by one ubiquitin, and this process is known as monoubiquitination. So it is very important that the substrate, the protein which has to be degraded, must be attached to at least 4 ubiquitin proteins so that they can be degraded. Okay, so now another ubiquitin is brought by this process, and here the lysine group of the ubiquitin is present. Okay, so another ubiquitin will come, that is the glycine of this ubiquitin will interact and form the bond, that is the isopeptide bond. So at least like this, 4 ubiquitin are attached, and this process is known as polyubic polyubiquitination. Okay, so to summarize this whole process of ubiquitination, we'll see this diagram. Okay, now you see here, this is the E1 enzyme, and it has the active cysteine sulfhydryl group here. This ubiquitin, with the help of ATP, attaches to this sulfhydryl cysteine group. Okay, and this releases the one molecule of AMP and the phosphate. Okay, this is the first step. Okay, this is the first step. In the second step, it involves the E2 enzyme. Here the E2 enzyme takes away the ubiquitin. This ubiquitin is taken away by the E2 enzyme and attached to this sulfhydryl cysteine site. This both involves the thioester bond. Okay, this is the second step. In the third step, the E3 enzyme, which is attached to the substrate, now the E2 enzyme comes and bind to this E3, and now this ubiquitin is taken by the amino group, that is the what you call the protein residue lysine, and this is the glycine. This is taken away by the lysine N terminal, and they both form the isopeptide bond. You see here the amino group and the ubiquitin, they are both attached to each other by the isopeptide bond. Now this E2 and E3 groups leave, and the substrate are then added with another ubiquitin protein. They are at least the 4 proteins, so that they can be degraded. They then go to the proteasome and be degraded smaller amino acids. So this is the whole process of ubiquitination. I hope you've understood. What's wrong with this? Okay, so I hope you've understood. Thank you.